Welcome back. So we've talked about the various structures of the cell itself, and really when we're looking at those things, not only are we talking about the cytoplasm and all the organelles within the cytoplasm, but we're also talking about the cell membrane, what is enclosing everything. And when we start to talk about the cell membrane, it's kind of something that we really dive deep into. So as much as the other organelles are very important, you see things like the chloroplasts and the mitochondria. These things are focused on because they are vital for energy in cells. But also the outside is very important in protection and also allowing the flow of molecules in and out of that specific cell. So every cell is bordered by what we call a cell membrane, or another phrase or another name for it is a plasma membrane. And the plasma membrane, it's made up of these two layers that come together, and they are made of phospholipids. So sometimes you'll hear it referred to as a phospholipid bilayer, bi meaning two. So it's two layers of phospholipids that create this cell membrane that is going to keep everything that you want inside and everything that you want outside to stay outside, and it's going to control what comes in and what goes out. So the functions include to hold the contents of the cell, takes in food and nutrients, aids in building and exporting the molecules. So not only is it taking in things, but it's making sure that you're getting rid of things, and it allows interactions with the environment and the neighboring cells, or the cells that are close to it, or the cells that make up that tissue. So we like to refer to the plasma membrane as the gatekeeper of the cell. It is what is saying what's going to be allowed in and what's going to be exported out. So when we talk about a phospholipid bilayer, well, it's made up of phospholipids. Okay, remember we talked about lipids, we talked about fats, oils, waxes, those are lipids. Well, this is a certain type where we talk about phospholipids, and it's two layers of these, so we call it a bilayer. Excuse me. Each layer has these components to it, these phospholipids, and the phospholipids are broken up into these two portions. So there's a top portion that is called the head, and then there's a bottom portion that's called the tails. And the head is actually hydrophilic, meaning water loving, meaning it's going to attract water. So that is always that top part of a phospholipid is going to always kind of pull water towards it, while you have the tails that are hydrophobic, and they are not attracted to the water. They are going to push water away from them. And when you look at a phospholipid bilayer, these bilayers, it's made of these little phospholipids, okay? And there's an outer portion, which is one layer, and an inner portion, which is the other layer. And they're actually facing opposite ways. So the heads on the outside are facing towards the outside, and it has the tails on the inside. And then on the inside layer, you have the heads facing towards the inside, and you have the tails on the facing towards the inside of that membrane. So the tails are all on the inside of the membrane, and the heads are either facing outwards or facing inwards from the cell. So that is what makes up our two layers. You see here, this is one layer, and this is another layer going all the way around. And when you look at the hydrophilic head, you have it's a glycerol backbone linked to these phosphorus-containing molecules, which is makes us think of a lipid that is made up of glycerol backbone with the fatty acids. And then you have the hydrophobic tails, which are composed of carbon and hydrogen chains. Now, with molecules, they are going to flow in and out based on what the cell membrane allows, and molecules are actually embedded within the plasma membrane to help it perform its function. So as much as molecules are part of what goes in and what goes out, there's actually more to the phospholipid bilayer than just the phospholipids. There's also other molecules that are part of it that play a role in the function of the cell membrane or the plasma membrane. So you have these transmembrane proteins, you have various lipids, you have carbohydrates attached as well, and you also have surface proteins. So with the hydrophobic and the hydrophilic forces, they're going to determine the orientation of the proteins in that cell membrane. Now, there's different functions of the different molecules that are there. You have these receptor proteins that are going to bind to external chemicals to regulate the process within the cell. So you have these special proteins, and they have special shapes, and they are responsible for certain chemicals. The shape really depends on which chemical they're responsible for. And then you are going to have these recognition proteins, which are kind of like a fingerprint or a flag for an identification by other cells to say, hey, I belong here, or in fact, I don't belong here. You have cholesterol as well, which helps maintain the flexibility. Carbohydrate chains as well are going to provide a fingerprint or a flag for identification. And then you have these transport proteins, which are going to allow molecules to move in and out of the cell. You also have membrane proteins, which are going to accelerate intracellular and extracellular reactions on the cell membrane. So where we see this in real life or in the human body is patients with cystic fibrosis. Cystic fibrosis, as much as we look at the varying or the various symptoms of someone that has cystic fibrosis, and you talk about issues with the lungs and mucus buildup, this is really a cellular disease, a disease that occurs on the cellular level because of these faulty membranes. So when the cells have a faulty membrane, you not only is it going to affect that cell, but it's going to have a bigger picture effect. So that's where you see people with cystic fibrosis. They're having mucus issues because of these faulty membranes, and because of those mucus issues, you're going to have patients that are going to have to use this thumping on their chest to loosen up the mucus, and they have special vests to help to keep their mucus issues at bay. So the thick and sticky mucus produced by someone with cystic fibrosis, it collects in the lungs, and it impairs the lung function. And this increase in mucus can lead to bacterial infection. So that's why people are going to have to use this thumping or these vests to make sure that they don't get sick. Also, you see when we talk about these proteins within the cell membrane, they have specific shapes to them for specific molecules, so that they have what we call receptors, and these receptors have specific shapes. And what happens is they are going to respond when that protein comes in contact with that molecule or that chemical. Now, what happens a lot of times is that we are able to use medicines to block that response. So you have things like adrenaline, which is going to make a cell respond in a certain way, and adrenaline has a certain shape to it, and it's going to, once it goes into that receptor, that beta receptor or that protein receptor, it's going to have a response. But what we are able to do is we're able to block that. So we're able to give a patient certain medicines, and those medicines have the same shape. They're going to block the adrenaline from going into that receptor and causing that response. So in stressful situations, you see the adrenal glands are going to pump out adrenaline. And adrenaline is the fight or flight chemical. It cannot enter the cell when you are on beta blockers, so that actually causes a decrease in anxiety. You're blocking that anxiety response by blocking those receptor proteins. Now, I said that they have these kind of flags to them, and they're important. Well, you want to, the human body, a bigger picture. So as much as we're talking about cells, we're also talking about tissues and organs and organ systems and an entire multicellular organism. Well, the entire multicellular organism, they have many different cells within them, and there's also many invaders, many pathogens, many things that want to interfere with you. So it's important for your body to be able to pick up on what's supposed to be here, what's not supposed to be here, and that is because of these fingerprints or these flags. And you see, if you have these flags, you're able to pick up on, within the body, you have these CD4 markers. So HIV infects the body by binding to these CD4 markers of these cells, and your body is now no longer able to function properly. So passive transport is what we usually talk about, where it's spontaneous diffusion. This word diffusion means free flow of molecules moving from an area of high concentration to an area of low concentration. So I always explain it like if you are at a party and there's 2 rooms to the party, and if you're in one room and it starts getting packed in there, well, what are you going to want to do? You're going to gravitate to the room with less people in it, as much as we, can say, well, I always like to, be talking to a lot of people. But just generally, if you are shoulder to shoulder and you are cramped up and you are uncomfortable, you're going to go where there's less people. You're going to go in that room where there's not a lot of people. Well, the same thing goes for molecules. So if they have the chance to move, and they're always moving, if they have the chance to go somewhere else, they are going to go somewhere else. So the one example is if you were to put some food dye in water, it's going to slowly diffuse throughout the water. Same thing goes with a sugar cube. If you put sugar into your water, it's going to diffuse throughout the water. It's not just going to stay a cube in that water. So that is the idea of diffusion. But when we see it in membranes, we see that it's crossing that membrane. So it's going to, if there's too much of it on the inside, it's going to move to the outside. If there's too much of it on the outside, it's going to move to the inside, if it's able to. Some molecules are too large, they cannot cross that barrier, but smaller molecules, water molecules, and smaller monosaccharides, smaller sugar molecules, those molecules are small. They're able to pass, and they're going to go from an area of high concentration to an area of low concentration. So we talk about 2 different types of diffusion. There is simple diffusion, where molecules pass directly through the membrane without the assistance of another molecule. But then you also have facilitated diffusion, where it's going to be facilitated, it's going to be helped. And these molecules move across the membrane with the help of a channel or a carrier molecule. Now, passive transport occurs when molecules are going to move across the membrane without energy. So now we're talking about passive versus active transport. So passive is going to use no energy. Active is going to need energy, need ATP. And within passive transport, there are 2 types. You have simple diffusion and you have facilitated diffusion. Now, one type of diffusion, so diffusion talks about the passive transport or movement of molecules, but when we're talking about not just any old molecule passively moving, but we're talking specifically about water molecules moving, we call this osmosis. So diffusion really is osmosis is diffusion, but it's talking specifically about the diffusion of water across a membrane. And there's different types of solutions. So when we have a solution where the solutes or the molecules are balanced, both inside and outside, because remember, we're talking about the amount of molecules on the inside or the outside. Remember, they move from an area of high to low. So if they are equal, there's not going to be a net or a major change in the amount of molecules. So we call that isotonic. Okay, it is balanced. So you're not going to see a big rush of molecules out. You're not going to see a big rush of molecules in. When you have a hypotonic solution, you're going to see the solute concentrations are lower in the extracellular fluid. Okay, so what's going to happen is water diffuses into the cell. So you're going to have more solutes on the inside. So you see these little specks, those are supposed to represent molecules, and there's not many of them on the outside. Well, what happens is these molecules are not able to diffuse, but what they can try to balance out is the amount of water in relation to the number. So if I say, hey, I only have 5 molecules on the outside compared to 5 molecules for this gallon of water on the outside, but on the inside, if I were to compare it to the gallon, I have way more. So the water is going to try to balance out really the relative amount of water, and it's going to rush in. So the water is saying, you know what, I have 5 molecules for X amount of water on the outside, but I have 30 molecules for X amount of water on the inside. So water says there needs to be more water on the inside because the ratio is off. So what happens is if you have a scenario where there's a hypotonic solution with red blood cells, water can actually rush into those red blood cells and make them burst, which causes an issue. So what you want to do when they give you IV fluids is they want to give you an isotonic solution. And our body fluid does have salt to it, so it's not just pure water. It does have salt to it. So an isotonic solution, an IV bag, does have salt in it. But you don't want too much salt. You don't want too little salt. So the flip side of hypotonic solution is a hypertonic solution, where you see the solute concentrations are higher in the extracellular fluid, the fluid on the outside. So here you see, oh, I have some molecules on the inside, but I have a lot on the outside. So water is going to pick up on that and say, hey, you know what? The ratio is off. I have way more water per molecule on the inside of the cell than I do on the outside of the cell, and the water is going to rush out. So when you see something like a plant molecule, it's going to rush out, shrivel up. Same thing goes for a blood cell. It's going to shrivel up and be useless and not able to function. So depending on what type of solution that a cell is put in is going to affect the flow of molecules, the flow of water, the osmosis that occurs. So we call this amount of solutes in a liquid is tonicity. All right, and tonicity is the relationship between the concentration of the solutes inside the cell and the solutes outside of the cell, and that tonicity is going to influence whether molecules are going to flow out or molecules are going to flow in. So you have the isotonic, which is balanced or equal, and then you have the hypotonic, where the concentration of the solutes outside the cell is lower than the concentration on the inside of the cell. So then the water is going to try to make up for it, and you're going to see that flow, opposed to hypertonic, where the concentration of the solutes outside of the cell is higher and the water is going to rush out. So osmosis in action, you see something like dehydrated celery contains many dissolved molecules relative to the water molecules, and that's why you see that it becomes dehydrated. When you place it in a distilled water that doesn't have any salt to it, you're going to see that the water is going to rush in, and that dehydrated celery is now going to be rehydrated because that water is rushing in to create that balance again. So we have passive transport, and then we also have active transport. So as much as there's simple diffusion and facilitated diffusion within that active transport, we also have the flip side of passive is active transport, and this uses energy. So energy is needed for this to occur in order for small molecules to move in or out of the cell. And you see that there are two types of active transport. You're going to have primary active transport, which directly uses ATP, and then you're going to have secondary active transport, where transport proteins are going to obtain energy in an indirect method, not a direct use of ATP. So keep these things in mind as we move forward. You want to have a firm grasp of not only what's going on with the cell membrane, but all the organelles in the cell.